Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Kia ora koutou katoa. Welcome to Sigest. I'm your host, Hilary Ireland. From banana peels and tough avocado skins to crisp apple peels and smooth nectarine skins, we see a huge range of fruit skin surfaces and textures. Why is there so much diversity? How does it arise, and can we change them to make some skins more edible? Today, we are joined by Research Associate Mikaela Douglas, who is exploring the science of fruit skin diversity. Welcome to the podcast, Mikaela.
Kia ora, thanks for having me here.
It's a pleasure to have you. Before we dive into the science, could you tell us a bit about yourself and your role here at Plant and Food Research?
Yes, well, as you said, I'm a research associate. I'm based in the new Cultivar Innovation portfolio, and I am part of the Accelerated Breeding Concepts team here at PFR Mount Albert.
That sounds cool.
Yeah, it is.
So you're studying the science of fruit skin diversity. Why study this aspect of plant biology?
There's two primary reasons I study it. For the consumer aspect of it, there's lots of consumer traits that you can find in the skin. It's the first thing a consumer sees is the very outer surface of the skin. It can tell you a lot about ripeness, about the quality of the fruit, and as well as that, kind of the experience you'll get eating the fruit. And secondly, it's important for production as well too. Some skins are better storers than others. Skin disorders as well too. Some fruit are more prone to different conditions of the skin, whether that be scabbing or wounding or even fungal infection or whatnot. It's also important to mitigate pathogen and pest attack too, as well as reduce biotic and abiotic stressors. A biotic stress is something that is of biological origin. So say pests like, bugs and whatnot, or even fungal attack or pathogen attack, even seed dispersers as well too. And an abiotic stress is something that is not of a biological origin, such as the changes to environment or to humidity.
It's doing a lot. It's a busy little organ.
It is, yeah. It really packs a punch for how small it is.
Yeah. Yes, cool. So in a previous episode of Sigest with Dr. Richard Esplee, he told us about research aiming to enhance the nutritional content of blueberries by filling the colour void of the flesh to match the colour of the skin. What is it about the fruit skin that makes it so nutritionally important?
There's some nutrients in the skin that may be in bigger quantities versus the flesh because they also indicate ripeness, or they're used as part of the defense mechanism that the skin provides. Things you'll find are antioxidants such as anthocyanin and other color compounds like carotenoids. There's also lots of fiber that help create the cell walls that keep it very impermeable. And there's also lots of vitamins, including vitamin A and vitamin E, and even folate too.
So no peeling the skins, eh?
If you can, I would recommend eat all your fruit with your skins.
Good advice. So sometimes the decision to eat a fruit skin is purely down to preference. For example, some cultures actually cook and eat the tough rind of things like bananas and pineapples and watermelons, if you can believe it. So are all fruit skins edible, really, in theory?
Most of them are, with some caveats. Yeah, that's really cool about the watermelon. I was actually watching a video the other day of someone making kimchi out of watermelon rind. I thought, how amazing.
Good.
Yeah, there's really poisons found in fruit skins. Like you can get this oily resin in mango skins, so definitely wouldn't recommend eating that, but it's very rare and you're more likely to get it in the peels of vegetables, especially root vegetables like green potatoes, damaged kumbra. You're not actually meant to eat parsnip skins unless they're cooked. If they're raw, they can be toxic to humans, but this is also rare. And MPI does monitor food residues, including pesticides. And it's very, very rare that we get tests that show above maximum residue limits, which we have set to much lower standards than food safety limits.
Why is there so much diversity in the fruit skins that we see?
Well, all these different fruit are grown in different climates in different parts of the world, so they all have to experience different challenges. And also, they have all co-evolved with the seed dispersers, particularly fleshy fruit, where the flesh of the fruit is the reward for the disperser. So, what you need to attract a bear versus a bird, maybe even a mice or a human, is different between each fruit. And when you're looking at different environments, some environments may be more hot, so they need to be able to create a microclimate on the surface of the skin to reduce that heat stress, or maybe they want to avoid losing lots of water, so they create a waxier, tougher skin as well too. But they change depending on the environment.
Okay, so what is the function of the fruit skin then?
The fruit skin primarily is to protect the fruit from both biotic and abiotic stressors. It's also very important to regulate fruit growth. It can help withstand pressure to changes in volume as the fruit expands over time. And also, it provides lots of good visual cues to seed dispersers about when the flesh is ready to eat. Like color pigments will accumulate as the fruit matures, and at that point, the seeds should be all mature and ready for dispersal.
So can you give us a high-level overview of the structure of a typical fruit skin?
There's so many different types of fruit skins. I kind of describe it as fruit skin lives in a continuum of phenotypes and cell types. The most extreme ends of this continuum, you have peridermal, or in a more simplified version, a dead skin. And on the other side, you have an epidermal skin or a live skin. Mm-hmm. And in between, you have intermediate phenotypes. For example, the peridermal dead skin is a skin type that is made of a periderm, which has also supported by a phallem and a phallogen, and it produces many layers of cork cells that are covered in this compound called suberin that will make it impermeable to water. And these cells, as they expand over time as the fruit develops, they will then go through programmed cell death and collapse into the russet patches that you're familiar with on an apple or a pear. You also can get hairs as well too on any skin type or other types of trichomes. Trichomes are outgrowths on the outer surface of skins. For example, you get hairs. You can also get lendisols, which are little spaces for gas exchange between the fruit and the environment. You can also get cracks, often just as the fruit develops and expands. And you can also get scales. And even furthermore, hairs can have different types of hairs. Some will be glandular, where they have a little bulb at the end of them that produce phenolics or aromatics that can either protect against pathogen or maybe create nice aromas, or be made of multiple cells or just single cell. On the other end, you have live skins, which are very, very simple in contrast. You simply just have a epidermis and a cuticle. The epidermis will support the strength of the cuticle and produce more wax, and a cuticle is just made of lots of very long-chain fatty acids, which will modulate drought stress, salt stress, all sorts of environmental stresses.
So the live skins are really simple.
Yes.
And do they have hairs and lenticels and things like that? No?
They do have some, but not to the same degree that you see with dead skins.
Right, okay. So talking to that continuum that you're talking about as well. So what are examples of live skin?
Some examples of live skins would be cherries, tomatoes, fruit that have very, very thin skins that you can eat whole without having to worry about, scooping out the flesh or cutting the rind off.
Right. And at the other end?
On the other end, some examples of dead skin include watermelon and rockmelon, especially, the webbing you get on the surface of them. Avocados are technically dead-skinned, but they still, produce lots of pigments that indicate ripeness as well, too.
So they're more further away from fully dead and still living in some aspects.
Yes.
Right, on this continuum.
Yes.
And what about the middle?
In the middle, you get different types of semi-russet, which, as opposed to the, developmental russet that you get on the whole surface of peridermal fruit, you get little parts that russet in response to environmental stress. So, say apples and pears and cherries, we get those little cracks or webbing on the surface, which could be a consequence of there's a droplet of water on that one portion of the apple for too long. And the fruit decides, no, I need to reduce the amount of water that's coming into the fruit.
I I see. And russet apples can be quite tasty. Is there any problem with eating russeted areas?
Oh, no, definitely not. There's more fiber and vitamin E in the russet, actually, so go for it. It may have a weird texture, but it's very good for you to eat.
Great. So do all fruit skins have all of these different types of modifications, the glandular trichomes, the lenticels, the other trichomes? Are they present on all fruit skin all the time?
Not all of them are on all fruit, and to varying degrees. For example, some dead skin fruit probably will have more lenticels because it's more impermeable to water and air, so it still needs spots for gas exchange, obviously, because even when you pick the fruit, the cells are still breathing. They need to get carbon dioxide out at some point. Like, for example, when you put a banana in a paper bag with another fruit and then it ripens overnight, the ethylene that's being emitted from the banana still could be being uptaken by the other fruit through these areas of gas exchange.
Very cool. And the glandular trichomes, are they common? Are they
not necessarily? You sometimes get them on the fruit skins, but you're more likely to get the non-glandular types on the fruit skins. The fruit skins have found other ways to protect against pathogen attack. And they also produce phenolics as well, too, that will have a secondary function of protecting against pathogen and pest attack anyways.
So there's a lot of variety there, a lot of diversity in fruit skins.
Yes.
What particularly is the aim of your research?
My research is looking at the genetic networks behind these fruit skins and figuring out what genes control the fate of a fruit skin to tell it, okay, we're going to have a dead/russet skin or an epidermal type or somewhere in between those. I'm looking specifically for transcription factors, so genes that control other genes and sit at the top of pathways that will then turn on biosynthetic genes that may start causing more wax to accumulate.
Okay, right. That sounds tough. With all those different skin fates, it sounds like there would be quite a complex regulatory network. Is it a simple on and off switch between edible versus inedible, or is it far more complex than that?
Yeah, it's quite complicated because a lot of the different fruit skin characteristics overlap with each other in some way. It seems to be that there's a bit of crosstalk between different pathways. Like, for example, if you reduce the wax on the surface of a fruit, you end up getting less hairs overall as well, too. And like part of my research is trying to figure out how are those connected? Is it one is a consequence of the other, or is it they're just controlled by the same genetic network or transcription factor? Or is it just a byproduct of reducing wax load? Like, for example, as well, when you have live skin fruit where they start to russet a little bit in response to environmental damage, there is a series of programmed cell death. For example, in russeted patches in apples and cherries, the cells of the epidermis de-differentiate, and instead you get basically a cork layer that will then develop lots and lots of cork cells that will then be superized and compressed. But what I'm trying to figure out is what transcription factors tell a fruit, oh, we've been damaged here. We need to turn on russeting and kill off all the epidermal cells so far and create a cork layer instead.
Wow. So when you talk about cork, do you mean like cork that people find in their wine bottles overseas?
Kind of. It's of the same cells, but just a little bit more suberin in it, and obviously not as big and thick as like the cork bottles from wine.
But it's the same sort of cellular structure?
Yes, same cellular structure. Yes.
Right. Okay, good. So you talk about wax and hair and some crosstalk there. Is that similar to the story that we hear about the difference between peach and nectarine?
Yes, as a matter of fact. Nectarine is a sport of peach, which means they're the same species apart from just a small genetic change that induces the nectarine skin phenotype or appearance. Specifically, in this case, there is one gene that has a big deletion of 1,200 base pairs, and that is enough to turn off the network that will produce wax on the surface of the skin. And as a consequence of reducing the wax on the surface, you have less hairs as well, too.
Right, which gives you the nectarine
Appearance.
phenotype.
Yes,
I
yes.
see. So the fuzziness of peach is through all of the hair development on the skin.
Yes, exactly.
Right. Okay. So how are you going about finding the genes when you've got a raft of genes probably in the genome and all these different fruit skin characteristics? How do you break it down and approach the question?
There's lots of different tools we can use to understand genetic network and understand why skin has adopted a certain fate. Normally, we start off with some sort of transcriptomics experiment, which means you're just comparing the overall gene expression in a certain tissue versus another tissue. So, for example, you could get russeted apple versus an apple that's not russeting and measure the overall gene expression and what genes are being expressed in those tissues at several time points and then compare them. And it should give you a good idea of what genes are being turned on when and where. We can also use a bit of microscopy and histology to identify the compounds as well, too. You can even use some chemistry techniques such as GCMS or UPLC to measure, fatty acids or folate or other proponents of secondary cell walls, too. We also use gene editing for understanding. So once we have a couple of candidate genes we think regulate different aspects, such as maybe a gene that controls the accumulation of wax on the surface of a live-skinned fruit, we will then insert it back into a model plant to overexpress the gene to see if we can get more expression of it, or we can use CRISPR-Cas9 editing to silence it as well, and perhaps reduce the overall load of the wax on the surface of the plant. We can also use breeding as well to kind of understand what's going on with the networks and the genes that control skin type, especially the dominance of them. For example, if you breed a peach and a nectarine, you get a kind of an intermediate phenotype, a peacharine.
Oh,
yeah.
And is that mostly fuzzy or?
It's partially fuzzy.
In patches or just
overall partially
a close
fuzzy?
Shave.
Yes. Yeah, close shave. Yeah, it's like a number 0 versus a number 1 haircut.
Okay. Wow, that's a lot of different methodologies. How's it going? And what have you found so far?
We're lucky we've been able to identify a couple of transcription factors that are good candidates for regulating aspects of hair and wax development. What we're trying to figure out is how they control these pathways. And it seems that there's crosstalk between them and the propensity to russet in some fruit as well, too. So if you turn on a switch that tells it, hey, we're going to produce more wax, there could be a response where the fruit produces less russet in response or more. For example, done some gene editing where we've created plants that have less, and fruit that have less hairs and wax, but they are still russeting. And when you look at the overall gene expression in those tissues, there is still some expression of russeting factors. So there's still a lot to uncover here. It's a very complex pathway, and all these genes seem to talk with each other.
Right. It sounds like it's not so much an on-off switch between living and dead skin.
No.
It's like, lots of different switches controlling different parts of the dead skin phenotype. Okay.
And you'd have to edit or breed out a lot of off switches in order to get your phenotype of choice. But
Okay. So quite a tricky aim then.
Yes.
Okay. Well, good luck. Good luck with it. And so what are the next steps of the research?
Yeah, well, we need to uncover more about this pathway. It feels like every time I've answered a question, I've just opened up Pandora's box and now have 10 more questions to ask and answer. But we've at least gotten some good candidates that are helping us to finally start unpicking this complex series of questions.
Yes, right. Well, hopefully we'll start to see new varieties with strange and interesting looking skin types.
That'd be cool.
Eventually in the future from your research. Lastly, what are you excited about for the future of your fruit skin research or others?
I am very interested in this for the health component of it. Fruit skins, particularly peridermal/dead skin types, are perceived to be very better and more difficult to chew than, say, a live skin, an epidermal skin. And I think we're missing out on a lot of nutrition by not eating the fruit skin. So if we can produce an easy, convenient snack where you can just eat the whole fruit, hopefully then people are eating the whole fruit and mean they're getting a better boost of nutrients every time they go to the supermarket.
And eliminate a bit of food waste.
Yes, exactly. And also there is some potential benefit to use of pesticides as well too.
Okay.
In the future with this.
How would that work?
Well, I'd hope that when we're developing fruit with better skins, it can also resist more pathogen attack better than, say, a live skin fruit would. Or even improve the storage time of a fruit as well.
So many applications, and it's an exciting area to be working in.
Yeah, there's a lot to consider. I'm looking forward to seeing where this takes me.
Wonderful. Well, thank you so much. It's been a pleasure talking with you today. Thanks for sharing your time, telling us about your research, and we look forward to hearing more about your work in future episodes.
My pleasure. Happy to talk.
And thank you out there for listening. If you'd like to know more about the topics covered on Sigest, check out the episode pages on the Plant and Food Research website. Please follow or subscribe to the podcast. You can also rate and review each episode or share it with a friend. Thank you, and mā te wā.